Good morning, student. So, in our today's video, we will talk about reproductive health, in which we will discuss about sexually transmitted disease, infertility, and various assisted reproductive technology or ART. So, let's start it. Medical termination of pregnancy. So, intentional or voluntary termination of pregnancy is called as a medical termination of pregnancy or induced abortion. MTPs or medical termination of pregnancy is mainly meant to get rid of unwanted pregnancy, either due to casual unprotected intercourse or failure of contraceptive used during the coitus. It has a significant role in decreasing population. So that means if someone wants to reduce the population, so medical termination of pregnancy is a useful method. This method is a safe within first trimester or up to 12 weeks of pregnancy. But the problem is that majority of the MTPs are performed illegally by the unqualified peoples, which are not only unsafe but could be fatal too. And frequently, if the fetus is found to be female, it is followed by MTP. That is totally illegal. Now we'll talk about the sexual transmitted disease. So the disease or infections which are transmitted during the sexual intercourse are called as a sexually transmitted disease. These sexually transmitted disease are also called as a venereal disease (VD) or reproductive tract infections or RTIs. Some common sexually transmitted disease are gonorrhea, syphilis, genital herpes, chlamydiasis, genital wart, trichomoniasis, hepatitis B, and HIV. Except hepatitis B, genital herpes, and HIV infections, others are curable because these are viral disease. So this is a list of some common sexually transmitted disease, their causative agent and detection technique. So the first sexual transmitted disease is chlamydiasis, and the causative agent is chlamydia trachomatis, and the detection techniques that is used for this sexual transmitted disease is the clinical and gram staining of the discharge detections and nucleic acid hybridization. The second disease is gonorrhea, and the causative agent is Neisseria gonorrhea, and detection techniques include gram staining of discharge and then culture of that causative agent. The third sexually transmitted disease is trichomoniasis, and the causative agent is Trichomonas vaginalis, and detection techniques as microscopic examinations and culture of that causative agent. The third is genital herpes, and the causative agent is herpes simplex virus, and detection techniques includes clinical antigen test and PCR, that is a polymerase chain reaction. The next sexually transmitted disease is syphilis, and causative agent is Treponema pallidum, and detection techniques include antibody detections and examples VDRLA, that is a venereal disease research laboratories. The next is genital wart, and the causative agent is human papilloma virus, and the detection techniques include clinical treatment, antibody detections, and culture and hybridization. The next one is hepatitis B and C. So for the hepatitis B, the causative agent is hepatitis B virus, and for the hepatitis C, the causative agent is hepatitis C virus, and for the detection of both diseases, ELISA techniques is used. The last one is AIDS, and causative agent is human immunodeficiency virus, and for the detection of this disease, ELISA and PCR techniques are used. So now we'll talk about the symptoms of the sexual transmitted disease. So it includes itching, fluid discharge, slight pain, swelling in the genital region. In female, it has been seen that these sexually transmitted disease doesn't show the symptoms at its initial stage and remain undetected for a longer time. And in later stage it may leads to the pelvic inflammatory diseases, which is also called as a PID, abortions, infertility, or even cancer in reproductive tract. Absence or less significant symptoms in the early stage of infection, and the social stigma attached to the sexual transmitted disease deter the infected persons from going for timely detections and proper treatment. If we talk about the preventions from these sexual transmitted disease, so the first thing is that avoiding sex with the unknown partners or multiple partners. The second is that always using condoms during the intercourse. The third is that in case of doubt, consulting a doctor for early detections. And the last one is that getting complete treatment for the diagnosed disease. That means if the person has been diagnosed that he has been suffering from some kinds of sexually transmitted disease, so he has to complete the course of his treatment. Now the next topic is infertility. When the couple are unable to produce children in spite of unprotected intercourse, then it is called as a infertility. And if we see the problem of infertility, so it may be in male or female. So these are some reasons of infertility. So this reason includes it may be because of physical reason, it may be of some kinds of diseases, it may be of some kinds of drugs or immunological or even psychological. So these are the some reasons which is responsible for the infertility. But in our society, often the female is blamed for a couple being childless. But more often than that not, the problem lies in the male partner. Now we will see some methods of infertility control. So these methods includes IVF ET. IVF, that means in vitro fertilization. ET, that means embryo transfer. The second is GIFT, that is a gamete intrafallopian transfer. The third one is ZIFT, that is a zygote intrafallopian transfer. And the fourth one is IUI, that is a intrauterine insemination. So one by one we will see all these methods in a. So the first method of infertility control is IVF ET, that means in vitro fertilization embryo transfer. So in IVF, fertilizations take place outside the body of female in almost similar conditions as that in the body, and which is followed by the embryo transfer is one of the such methods. So in IVF, female is induced to produce multiple egg or ova. Then egg is collected from the wife or donor, and the sperm is collected from the husband or donor, and then it is incubated in culture medium. Because of that, fertilization and zygote formations take place, and after that it is a transfer to the uterus of the wife, and where it will get implants and the pregnancy will be continue. Now the type of IVF. So basically there are the two type of IVF. The first type is ZIFT, that is a zygote intrafallopian transfer, and the second is IUT, that is a intrauterine transfer. So in case of zygote intrafallopian transfer, zygote or embryo with the 8 blastomere transfers to the fallopian tube. And in case of intrauterine transfer, embryo is transferred at the 32-celled stage to the uterus. Or we can also say that the embryo with more than 8 blastomeres are transferred into the uterus in case of intrauterine transfer to complete its further development. So this is the pictorial presentation of in vitro fertilization. So the first thing that happens is the woman is given hormone treatments to stimulate egg production. That means firstly we need to give the hormonal treatment to the woman for the production of egg. And the second is multiple eggs are taken from the woman's ovary. That means after the hormonal treatment so many eggs are produced, then we will take those eggs from the ovary. Now the third is, in the lab, the eggs are mixed with the men's sperm cells in a culture dish to become fertilized. That means after taking the eggs from the ovary of females and sperm cells from the male, then we will fertilize the sperm and egg in a culture dish. Then this fertilized egg or embryo are placed in an incubator for 48 hours. And after this, embryo are implanted in the woman's uterus or frozen for the future implanting. So if we talk about the significance of this in vitro fertilization, so first thing is that it's a boon to the infertile mothers. Men with the oligospermia, that means the low sperm counts, they can also use this techniques, and embryo can be frozen and used for the future. Now the second, assisted reproductive technology or ART, is gamete intrafallopian transfer. So in case of gamete intrafallopian transfer, we need to collect the ovum from a donor and transfer to a woman who cannot produce one but provide suitable environment for the fertilization. Sperm and ova are transferred to the ampulla of the fallopian tube with the help of the laparoscope, where the fertilizations and cleavage take place. So this condition is very useful when the fimbriae fail to capture the ovum and females having sperm antibodies in their cervical secretion. That means if the females will have a sperm antibody, so whenever the sperms will get entered, it will be destroyed. So in these conditions, gamete intrafallopian transfer is very. Now the next assisted reproductive technology is intracytoplasmic sperm injection. So in this condition, sperm is directly injected into the ovum in culture medium, then zygote or embryo formations take place, and which is a transfer to the fallopian tube or uterus. So here we can see the picture of intracytoplasmic sperm injection. So in this technique, firstly we need to hold the egg with the help of a micropipette, which is a 60 micron wide. Then we will take a healthy sperm with the help of a smaller pipette, which is a 7 micron wide. Then with the help of the micropipette, the sperm is carefully inserted into the egg. Then pipette is withdrawn, and then fertilization of egg and sperm take place, and then fertilized egg is implanted in the woman's uterus. Now the last assisted reproductive technology is artificial insemination. So this artificial insemination is useful in the case either the male partner unable to inseminate the female or very low sperm counts, which is called as a oligospermia. So in this technique, semen of a male partner or donor is collected, which is concentrated and then introduced into the vagina or uterus of a female. Then it will be called as a intrauterine insemination. All these techniques require extremely high care and handling by specialized professional and expensive instrumentations. Although by using all these techniques we can prevent the infertility, but there are some drawbacks which are as follow. The first drawback is these techniques are not useful with a female with damaged uterine wall. Second is it requires high specialized professional and expensive instrumentations, so it is available in few centers and available to few peoples only. And it raised several ethical, emotional, religious and moral issues in the societies. So these are some drawbacks of these techniques. But our law also permit the legal adoption of orphan child. This is one of the best methods for the couples who is looking for the parenthood.